Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about insertional inactivation. So at first, what is insertional inactivation? So it is a type of molecular biological technique basically used to identify recombinant DNA. So at first, what is recombinant DNA? Recombinant DNA is the combined DNA and combination of DNAs from different sources. So this insertional inactivation technique, insertional inactivation technique basically used to identify this recombinant DNA. So how it works? It works by disrupting the function of a specific gene. So generally we insert our desired piece of DNA in the plasmid. So suppose it is a plasmid. So here we introduce our desired piece of DNA in the plasmid. So it disrupts the function of a specific gene, okay, through insertion of that foreign DNA into its coding sequence. So it is basically a screening method to distinguish recombinant bacterial colonies and non-recombinant bacterial colonies. So what is the core mechanism? So now what is the mechanism behind this insertional inactivation? At first, remember one term, gene disruption. The foreign gene of interest is inserted into specific site within a selectable marker gene like antibiotic resistant gene or a reporter gene on a vector such as a plasmid. So when we disrupt or disturb or simply remove that particular gene, so in that way we can screen or identify the recombinant DNA. Another thing is loss of function. So the physical presence of insert or foreign DNA interrupts the original DNA sequence, rendering the marker gene non-functional and preventing the expression of its characteristic trait. For example, color production or showing resistance towards a specific antibiotic. So this is the mechanism behind insertional inactivation. We inactivate a particular piece of DNA by inserting our desired piece of DNA which replaces the original gene sequence. So there are different techniques to screen or identify recombinants. So the first one is blue-white screening. So it is a molecular biology technique used to identify recombinant bacteria, and this screening is based on color. Okay. So generally some plasmids have LAC-Z gene or LAC-Z reporter gene which produce beta galactosidase. Okay. So which produce beta galactosidase on the X gull medium, on the culture plate containing X gull medium plus IPTG. IPTG is an inducer that triggers LAC-Z promoter to ensure expression. Then on X gull medium, the bacteria having plasmid with LAC-Z will produce blue color colonies. Blue color colonies. Okay. When we insert our desired piece of gene in the place of LAC-Z gene, okay, replacing LAC-Z gene, it will produce white colonies, okay, instead of blue colonies. When blue colonies appear on the culture plate, it shows that all the bacterial cells are non-recombinant, having functional lac-Z gene. But when white color bacterial colonies appear on the culture plate, it shows all the bacterial cells are recombinant cells and lac-Z gene is inactivated by insertion or insertional inactivation. So some plasmids also have antibiotic resistance genes which is resistant towards a particular antibiotic. For example, plasmid PBR322. So PBR322 plasmid has two resistance genes, ampicillin and tetracycline. Resistance towards ampicillin and resistance towards tetracycline. When we insert our foreign gene in the place of tetracycline resistance gene, it will not show resistance towards tetracycline antibiotic further. So the recombinant cell lose their resistance towards tetracycline but remain resistant to ampicillin. Okay, so suppose this is PBR322. So it has gene tetracycline resistance gene as well as ampicillin resistance gene. Suppose we insert our gene of interest here in place of tetracycline resistance gene. It will not show resistance towards tetracycline further. That means recombinant cells lose their resistance to tetracycline, but it remain resistant to ampicillin because ampicillin resistance gene is already there. When we place our gene of interest in the place of ampicillin resistance gene, it will not show resistance towards ampicillin. So this is the case of insertional inactivation when we place our gene of interest or foreign gene in the place of antibiotic resistance gene. So with this I am concluding my lecture video here. It is all about your insertional inactivation. If you like this video, please hit the like button, share with your other friends and subscribe to my channel Sunanda's Tutorial.